Christophe Bernard. He was my PI when I was a PhD student. So you have him to thank or to blame. He's a team leader of a team called PhysioNet at the Institute of Systems Neuroscience in Marseille, France, down there. How many people is the team now?
25.
25, so a good sized team, a part of a larger neuroscience institute. And he's worked on stuff from dendrites, epilepsies, brain rhythms, bioelectrics, and opera. So he has a wide range of skills. So take it away, Christophe.
Well, thanks for giving me the opportunity to speak here. And I'm going to ask more questions than give you answers. And the topic is time, in fact, and things we do not consider when we do research in pathologies in the human body. And I will use epilepsy, which is my main topic of research, as an example, but what I'm going to say is valid for every other organ or diseases in the body. So this woman is extremely scared. It's not because she just heard the fake arguments between Elon Musk and Donald Trump during the last debate, but she has a seizure. And the seizure expresses itself with this intense fear reaction. And that's how seizures were described, and epilepsy was defined as a behavioral manifestation. So that's something that you see. So in many diseases, historically, it's the patient telling us that they feel sick or they are in pain or whatever. So when they go to a doctor and they try to find a solution. Now, in epilepsy, it's very different because when you have a seizure, usually you forget that you had a seizure. If you lose consciousness, you wake up and you don't know why you're lying on the floor. So it's the others who are describing to the doctor, or the doctor is witnessing it, what the patient has had. So basically, it's an observation. Okay? Like for any disease, it's an observation. So in addition to the behavior, with the advent of electrophysiology and the recordings of EEGs, we had a glimpse on what was happening in the brain. And you know that in the brain, there are lots of movement of ions, and you can capture the resulting movement of ions that go through the membrane as changes in the electric field. That's what we see. Now, each line is a recording site in the patient of the brain. And when the seizure starts, behaviorally, at the electrophysiological level, this is what you see. It's kind of big activity, high frequency, that is starting here, and it propagates also to other regions at the top, and even here. So a seizure at the electrophysiological level can be identified with that kind of recording by a specific signature. Now, just to give you some important numbers, epilepsy affects 1 to 2% of the world population. This is the second most common neurological disorder after migraine. And there are many types of epilepsies. The one we are working on is temporal epilepsy, which is the most frequent form in adults. And unfortunately for patients, this is one of the most drug resistant. Okay, so the question, of course, is to try to understand how these seizures are generated to treat patients. Now, why is this patient implanted with X electrodes that go in the brain in different regions? It's because she's drug resistant, and her only hope is a neurosurgery to remove the brain regions which are responsible for generating this activity and to try to make her seizure free. So we have a problem in epilepsy, is that drug resistance is extremely damaging for the patients and the families. And in the United States, it's estimated that the cost per year of a patient with drug-resistant epilepsy is $500,000 per person per year, all included. Okay, that includes also taking care of the patient and everything. So It's highly costly to the society. It's really damaging to the patient. So we have to find a solution. Now we have a big problem. Epilepsy is maybe the oldest field in neurological disorders to have been investigated at the scientific level. It was the first brain disorder to be recognized as organic, which means that you may try to find drugs to treat patients. As a comparison, Parkinson's disease and Alzheimer's disease, they didn't have that name at that time. They were not recognized as organic diseases, but as psychiatric disorder, and you needed a psychoanalyst or psychiatrist to treat the disorder. And we're not with drugs, but acting on the brain. But epilepsy is very old. It's the oldest field. And the first anti-epileptic drug to treat patients was introduced in 1848. That tells you that we have a long time history. And in 1885, Gowers, a major clinician, wrote a book, and he did like maybe the first clinical study in epilepsy, and he said, well, potassium bromide, this drug, is very useful. It can treat many of my patients, except 30% are drug resistant. That was 1848. Now it's 2024. We have 16 different types of molecules to treat patients. Drug resistance is 30%. We have not made any progress at all in our understanding of epilepsy. And the major problem when we try to understand epilepsy is so we try to explain this. So we have two possibilities. We use control tissue, and we can force a seizure to occur in the tissue, in vitro, ex vivo, or in vivo. And we record neurons, and we try to understand what is happening at the level of the circuitry. And if we, with the idea that if we understand what is happening, how they start, then we try to find a way to block them. Or we use genetic models or animal models of experimental epilepsy, which display spontaneous seizures like patients. And again, we compare the epilepsy with the control conditions, try to find differences, and by trying to find differences, then we try to infer mechanisms. This is true also for any disease. In any disease, what people usually do, they take a disease organ or disease condition, and they compare it to the control, normal condition, try to find differences, and infer mechanisms and treatments. And that's how we have been doing epilepsy research for decades. But the problem with epilepsy is the following. This is not a disease. Because seizures are occurring at random times. We don't know when they occur, why they occur, and when they are going to occur. It's not possible to predict them. Now, when you have Alzheimer's disease, it's a chronic condition. It's always there. It's not like 1 hour you have Alzheimer's disease, and the next hour you don't have Alzheimer's disease. No, you have Alzheimer's disease. It's a chronic condition. Epilepsy, you have a seizure, it's going to last 30 seconds, and then the next one will occur in 2.5 months, and you don't know why. It's like a random event. And not only that, it has been discovered that from flies to humans, if you push a neuronal network strong enough, it will seize. That has a very important consequence, is that any normal tissue can have a seizure in any species, which means that it's not a pathological activity, it's a physiological activity. You can have it expressed in any neuronal circuit, just have to push the system hard enough. What is pathological is to have a seizure because it can endanger a patient, but otherwise it's a physiolocal level, not a pathological level. The disease is because they occur and that can endanger the patient. So, but nobody thinks in that terms. They think it's a pathological state. It's a physiological activity that is triggered in some instances. In fact, any one of us has a probability of 1 out of 17 to have one seizure during our lifetime. 1 out of 17, that's a lot, but you may only have one seizure, maybe because of stress or lack of sleep,
And that's it. It's just an activity that the brain can generate, any brain can generate. So the goal is to better understand how to treat these epilepsies. But why do we have this lack of success? Because if you want to adopt the strategy of comparing an epileptic tissue with a control tissue, or what is happening before seizures is occurring, you cannot predict it. If you want to do proteomics or transcriptomics in the tissue just before the seizure, because that's where something should happen in the circuit, well, the seizures, they occur randomly in time, and there's no way to predict the next one. So when we kill an animal to do proteomics, for example, it can be here, but here it's the normal, the brain is normal. There's nothing really weird happening during that time. What is most interesting, maybe, is just to look before the seizure happens, but we don't know how to do that. So what we end up is comparing a tissue that is coming from an epileptic animal or from a human patient after neurosurgery and compare it to a healthy state. But we have no idea whether what we get when we isolate that type of tissue, whether it's meaningful to epilepsy, because these events are really transient. So that's a major difficulty. So we want to understand seizure mechanisms, but we may want to understand seizure risk. What if, instead of looking at mechanisms, there was some kind of underlying rule that would detect that at some time point during on this time axis, there is a higher probability to have a seizure for some reason. Now, this is seizure risk or seizure probability, and what governs seizure probability or seizure risk may be totally different from the seizure mechanism itself, how seizure starts. It's like when you have diabetes, you have favoring conditions that increase the risk to have diabetes, and these favoring conditions are very different from the mechanism of diabetes itself. So there are two parameters on which we could act, mechanisms and risk. Now, 99.99% of the research has been done on mechanisms, nothing on seizure risk, because we didn't know much, although we knew a lot, as you will see. So the problem is that seizures are probabilistic physiological events. They are physiological, they can occur in any brain, and they're probabilistic. So far, we cannot predict them. But as you will see, the probability follows some basic rules. And what I'm going to talk about is that the temporal structure of epilepsy unfolds over multiple time scales. And I will focus on circadian cycles and sleep-wake cycles and multidien. And as you know, circadian cycle is, the circadian rhythmicity is present, I think, in all living organisms on the planet. I don't know any organism on the planet where there is no circadian regulation of the expression of genes and protein. And we will see another form, longer term, multiple days that exists, at least in humans and rodents. But first, I would like to give you a hint of the general philosophy that we have tried to develop. In brain research, we don't have a conceptual framework. We don't have a theory of brain, how the brain works. Okay? I think it's in biology, it's true also. There's no theory of biology. We still don't know, I guess, how a single cell is working. There's no framework to understand how it works. So if we want to understand why the system does not work in epilepsy, we have no framework. So we cannot relate to anything, to any theory. So we are working blindly. Now, if we could have some basic rules, mathematical rules grounded into mathematics that describe the phenomenon, that could be tremendously helpful. As soon as you can write a theory or mathematical equations describing your system, you start to better understand your system and you can start to ask questions. Now, just to go back to seizures, this is what they look like on the EEG. This is like changes in ion flux across cell membranes. And the seizure starts here. That's how the clinicians define it, and ends here. And at some time during my career, I had been struggling to understand what this was. And the idea was the following. If we find a mathematical description of this event, can we use this mathematical description to produce some useful knowledge? But that will be the second part. So seizures, as I said, they are rare events, probabilistic events, but in most patients with epilepsy, there is another form of activity that is detected quasi-continuously in patients in the form of these large events, which we call them spikes. These big events, brief events, are not present in the normal population. Okay, so now to give you a gist of what I'm going to talk about, I'm going to show you a paper that was published in the Lancet in 1929, and that was totally forgotten for decades. Again, seizures to us, clinicians and researchers, are probabilistic events. We cannot predict them. And it's like there's no rule. But in 1929, in England, people with epilepsy, some of them were living in colonies by their families and taken care of by nurses and doctors in small villages that were constructed on this purpose. And they looked at the distribution of seizures in all these patients. And they found that some patients, so this is the percentage of seizures, and this is time of the day, from noon to noon. You have bedtime and waking up time of day. Many patients, they have seizures only just after they go to bed. Many patients have seizures only before they wake up. And other patients have their seizures only early afternoon and late afternoon. And they were describing that patients had a circadian or night and day increased probability to have a seizure at specific times during the night and day cycle. So it's not like it's random. No, it's patient specific, but it follows some strict rules. Now we had to wait nearly a century until in Australia they could record during many months and years patients living at home with drug-resistant epilepsy, and they had a system able to record their seizures and these interictal spikes. And this is here one patient. This is the time of the day again. And this is the number of days in the y-axis, and the black dots are seizures. You can see that this patient, during nearly 2 years, has seizures around 2 p.m. Okay, it's more complicated because that's biology. If you look at patient S10, obviously there's a change during the history of the patient with a shift to other time periods during which they have increased probability to have a seizure. So there is a circadian regulation of seizure. They are not random. They appear to follow specific rules. Which rules? We don't know, but they exist. And this was totally forgotten. And that was the first insight that there was something deeply rooted as a biological mechanism to explain why seizures occur, at least at certain time. And this is for seizure risk. Even better, that was totally discarded was, it's stuck. Okay. So in 1938, they found in the same colonies that some patients, true, they had seizures roughly at the same time of the day, but these seizures would occur in clusters. So this is weeks now, and this is the number of seizures which is displayed. And you can see that this patient has seizures regularly, nearly every 3 days. Bursts of seizures. Yes, every 2 weeks.
Now you have a different interval every month for females and males, so it's not related to the cycle. And here it was like every 3 months and a half. You can see it's really regular. So we all know about the circadian rhythm, but obviously, at least in patients, there is a multidien rhythm that is patient-specific, and there is a biological phenomenon that is driving the probability to have a seizure. It's increasing the probability to have a seizure every X days, and that's called a multidien rhythm. So, are there basic rules? As we will see about a seizure, we will see some basic rules, and basic rules that there is a circadian rhythmicity in epilepsy. It's not entirely random, and maybe we can use this. And it is, we will see that it's an hypothesis that it's linked to the circadian molecular oscillations that is present in all living organisms on the planet. I will show you briefly the hypothesis that I proposed. Then I will jump to this new concept, which was rediscovered in patients a few years ago, of multidien rhythmicity. That is a totally new biological rhythm that we know nothing about. So just as a quick example, just to show you why we think it's important to have some kind of framework, theoretical framework and mathematics behind what we are doing. So at some point in my career, I was despairing to find mechanisms, and then I asked myself the question that I should have asked from the very beginning, but what is a seizure? People tell us, well, seizure is this, is that, but what is this object? And I contacted someone specialized in nonlinear dynamics and bifurcation theory. And because my background is theoretical physics, I had the intuition that there were tools that could be used coming from nonlinear systems, because we are dealing with a nonlinear system, the brain. And it was Victor Giersa, and I went to see him. He had never seen a seizure in his life, and I asked him, "Can you help us to understand what is a seizure?" And he said, "Show me a seizure." I showed him this seizure, and so it's very easy. In 2 weeks, you will have the equations, and sure enough, 2 weeks later, we had equations. So I don't want to bore you with this, but you only need 5 differential equations to fully describe a seizure in terms of dynamics. Now, any time-dependent phenomenon in nature that can be described with a very small number of differential equations means that you have a core phenomenon. That's a basic point. There's a theorem that says that any time-dependent phenomenon can be described by a finite number of differential equations, coupled linearly or non-linearly. But here, 5 differential equations, that's the first evidence in biology that you can describe a phenomenon with so few equations. Okay, sorry, not biology, in the brain. So we were surprised, and it looked like we had stumbled upon something major and basic. And in fact, we derived from these equations a set of a theory and a classification of seizures. And this classification is based on dynamics. It's not based on biophysics. It's just describing what the signal is doing independently of the amplitude and the frequency of the signal. It's just describing how it's changing in time. Now, it's based on, it's an extension of Izikiewicz's work on neurons, on bursters, and we extended it to seizures. Bottom line is that we can identify 4 types of onset and 4 types of offset for seizures, which make 16 types of seizures. Now, these 16 types have been, we have found them in patients with epilepsy. We have the recordings, so we checked thousands and thousands of seizures, and we can find these 16 types of seizures. So this mathematical formulation gives rise to the first, in fact, rigorous mathematical classification of seizures. So far, the classification is totally empirical. So this is the first one that is mathematically grounded and verified in patients, and also
across all animals with epilepsy, flies, zebrafish, mice rats, monkeys, and of course humans. Having said that, this is just descriptive. It doesn't tell you anything about mechanisms. It's totally unrelated to biophysical mechanisms. So it's just helpful to describe what we see with the EEG. Just, we used it, in fact, to help patients. So in the lab, Victor has is a co-inventor of what is called the virtual brain. The virtual brain is a platform which is open access. And basically what you do is you scan the brain of someone. You get the anatomical MRI. There is another component where you can extract DTI, so how the system is, how the regions are connected to each other. These are big axonal tracts. From there, you construct an avatar of the brain of this individual, so with all the regions and how they are connected to each other. And then you can introduce the mathematical formulation, this model that we have described. And then in silico, you can get an avatar of a patient with epilepsy. So if you take a patient with drug-resistant epilepsy, you can virtualize their brain, of course, and model seizure genesis and propagation. Now, the cool thing about in silico approaches is that you can remove regions after region until you find the best set of regions to remove to cure the patient. So that's what we did. That's a clinical trial that lasted 2 years, multi-center. Nearly all the centers are in France, and we will soon know the results. The idea is that we gave this information from the virtual brain to the clinicians, and based on the recommendation, you should remove these regions. That is what is predicted, and what they did, we will see whether we can improve patients' neurosurgery outcome. And this was done, of course, because there was the virtual brain, but because also there is this mathematical formulation that fully describes dynamics of seizures. And without that, we couldn't have done the rest. So perhaps what we did, this nice, simple mathematical formulation will help cure people in the future. All right, so bottom line is that seizures, which are physiological phenomena, they follow simple rules at the minute time scale, which is the time scale of the seizure. So it's a built-in activity, again, in all organisms with neurons. And you can even evoke in a single neuron a seizure-like event. So with just a few ion channels, you can fully reproduce the same pattern, and you will find the same onset and offset bifurcations at the single neuronal scale. So it's like for me, a core mechanism. It's a very simple mathematical object. We can create a classification of seizures based on their dynamics. And again, this is the first scientific classification. Doesn't help treating patients, but at least it's useful to describe the phenomenon and maybe useful to help neurosurgery in the future. The problem is that there are many possible mechanisms to trigger a seizure. So this is just the beginning. Any type of this 16, there are many mechanisms that can account for that. Okay, so now let's switch to the circadian rhythmicity. So this is another example of patients with mesial temporal of epilepsy, and that's the distribution of seizures across the night and day cycle. And you can see that there's a higher probability in the afternoon. This is a rat model, and again, it's the same distribution. Now that's weird because the rat are nocturnal animals, we are diurnal animals, and we have the same probability at the same time. Why? This is again a big mystery, but that's a fact. But again, I want to insist on that. This is a probability to have a seizure, let's say, in the late afternoon, but it's not every day. Remember this diagram of the Multidien rhythmicity. So there may be during 4 consecutive days seizures at roughly the same time, but then you have many days with not any seizure. So it's just a probability. Doesn't mean that if you have a circadian regulation of your seizures, that you will have every day a seizure at a certain time point. But when you have it during this time course, it will be roughly at the same time. So it's circadian, yes, but with a probability. Now, how can we explain the circadian rhythm? There are lots of bacteria. There's a famous example of, I think it's cyanobacteria living in the reef. And they isolated 3 proteins from this organism. And so it's a single cell. And so one protein can autophosphorylate and dephosphorylate, and other 2 help the autophosphorylation or help the dephosphorylation. You take these 3 proteins, put them into a test tube with ATP, and the phosphorylation is oscillating in 24 hours. So the circadian rhythmicity is deeply rooted in, well, I don't know any example with no circadian rhythmicity of a single cell or an animal. So it's because the Earth is turning in 24 hours, and somehow it's been imprinted in all living organisms. Now we're not exception, but we have a central regulator of circadian rhythms. So circadian rhythms in humans, they are found in every organ. Even some organs are autonomous with their circadian rhythm. In the gut, for example, the circadian rhythm is autonomous, doesn't depend upon the brain. It can be resynchronized by the brain, but it can go freely by itself. We have circadian rhythmicity in the heart, in the stomach, mainly organs. In the brain, there's a structure called the suprachiasmatic nucleus. That's where it was evidenced, the mechanisms. And in their neurons, you have a set of genes which oscillate during the night and day cycle. So that's the transcript levels, that's clock gene. And this is a PER gene, which is an antiphase. And these are transcription factors, and they're going to change the expression of hundreds and hundreds of other genes, downstream genes, and that will upregulate their production or downregulate their production. As a result, the molecular architecture, the protein architecture of the suprachiasmatic nucleus is constantly changing from one moment to the next based on this oscillatory pattern of the transcription factors. This is reset every day with light when we wake up. It's resynchronized. This is the clock in the suprachiasmatic nucleus. But this clock is also driving what is called ancillary clocks, which are in all other organs and other brain regions. Bottom line here, in the suprachiasmatic nucleus, you have hundreds and hundreds of genes and proteins which are oscillating during the night and day cycle. And it was believed that it was only true in the suprachiasmatic nucleus, but not in other brain regions. So what we did was to look at the hippocampus, one brain region, doesn't matter what it does. It's important for us in epilepsy because that's one region that is very important from temporal epilepsy that is affected. Now this is color-coded. The expression, overexpression is in red and blue expression is low. And these are different side givers. So side giver 0 is when we switch on the light in the animal facility, and that covers 24 hours. And you can see that many genes follow circadian patterns as pattern in the hippocampus. So many genes and proteins oscillate during the night and day cycle. Now, this was like a shock because when we do research outside the suprachiasmatic nucleus, we don't care about the circadian rhythm. We study an animal, and we do it at time t, whatever that is. And maybe the next day we even use the same animal, but instead of the morning, we study the animal in the afternoon because there was no time to do it in the morning, and we average everything. But the structure is completely different between the morning. The molecular architecture is completely different between the morning and the afternoon. But nobody pays attention to that. So that was quite a surprise. And there were 2 papers in Science by the groups of Brunig and Brown. who showed that 70%, 70, of the proteins of the synapses in the forebrain oscillate during the night and day cycle. So it's not like minor. It's like everything is changing continuously in the brain. Okay. All right, so we have done it in the hippocampus, in the control animals, and what is happening in epilepsy? Well, in epilepsy, it's also oscillating. There are even more genes oscillating, but not the same. So now we have a different system in epilepsy where the molecular architecture is oscillating, but in a totally different manner. And maybe it's not even the same circuit anymore, because these are totally different sets of genes. And that poses a major issue in terms of interpretation of data, because as I told you, in any field dealing with diseases, we compare control and the disease state. And what many people have been doing for decades is to control expression levels of genes or proteins. And if there is a difference, the conclusion is usually, well, this may play a role to explain the disease because this gene is downregulated or this gene is upregulated. Now let's have a look at the alpha-3 subunit of the GABA receptor, one of the genes. It's important for the brain because it controls inhibition. So in the control, this gene is oscillating in a circadian manner. It's also oscillating in a circadian manner in epilepsy, but in a different manner. Now, if I were to do the experiment as we were doing before, and I was careful to kill the animal at the same time in both conditions, okay, I killed the animal at 11 a.m. Okay, I compare the control and epilepsy and say, oh, the gene is upregulated at 11 a.m. This may explain epilepsy because it's upregulated for some reason. But if I kill the animals at 3 p.m., then I get a downregulation. So now what do you conclude? Is it even valid to compare control and disease? Because if things have changed time dependent, time of the day dependent, doesn't make sense. There's no conclusion that you can reach. It's downregulated, upregulated at specific times. Yeah. But then what do you conclude from that? So this is a problem for data interpretation for everything that has been published with Western blots and RT-PCR or single-cell RNA-seq, whatever, you name it. If the genes and the proteins that you are studying are regulated in a circadian manner, well, if you don't take into account the circadian rhythmicity, that make your interpretation really complicated. So second set of conclusions, the molecular architecture of the hippocampus is cycling in control animals. It's true also in most brain regions. Well, in all brain regions studied specifically so far, it's true as well. We don't know another example, an example of a brain region that is not changing during the night and day cycle. The cycling rules are very different in epilepsy as compared to control, which raises a problem for data interpretation. And the question is, what are the functional consequences of the fact that we have a change in this cyclicity of proteins and transcripts? And that leads to a very down-to-earth and very simple hypothesis. If you put the two observations, the circadian rhythmicity of seizures and the circadian rhythmicity of the molecular architecture of networks, if you put them together, well, maybe it's very simple. As the molecular architecture is changing, for example, you have like a lot of expression of these genes at 9 a.m. and at 3 p.m. very low. Let's say that metabolism is super efficient to produce ATP at 3 p.m. and not efficient a lot in astrocyte because I changed the cell because it must not be only neurons. And let's say that you have like 4 proteins coding for receptors at the synapses at 3 p.m. and only 2, at 9 a.m. and only 2 at 3 p.m. So that's the molecular architecture that is changing from one instant to the next. And maybe at some time, the network is close to seizure threshold. It's in a more excitable state, and maybe it's easier to have a seizure at that time.
So here I plotted like network activity and here seizure threshold. And maybe what the brain is doing is following this circadian rhythmicity, periodically you get close to the threshold at around the same time of the day, but you remain below most of the time. And there are other factors like the multidien rhythms that allow the brain to, these regions to cross the threshold and express seizures. So that's a very basic and simple hypothesis that could maybe provide us with a framework to try to understand why seizures occur when they occur. Now, we have no idea what may cause this, of course, but this is kind of a framework that we could work upon.
Okay. So this conclusion is that the molecular oscillations that are underlying the changing architecture of the circuits may be sufficient to explain the circadian rhythmicity of seizures. They increase the probability, but the other factors must come in to make the seizure occur every whatever day. And again, there are many paths to seizures. So this probability may depend upon the type of seizure and the threshold itself, how it is determined. There are many rules, biophysical rules behind that, which remain to be explored. And because the brain regions, they oscillate with their own molecular pattern. What is happening in the hippocampus in terms of molecular oscillations is totally different from the suprachiasmatic nucleus, totally different from the cerebellum. So if you have seizures originating from the hippocampus, they may follow different rules in terms of circadian rhythmicity as seizures coming from the somatosensory cortex, for example, which is driven by a different oscillatory architecture. So that complexifies our work. Now, to finish, let's look at this new rhythm, this multidien rhythm. So how was it identified? Well, you may remember that there are two types of pathological activities in the brain, seizures, and here, these interictal spikes. Now, in the US, there is a device that is implanted in patients that has been implanted for decades now, which is called NeuroPace. And NeuroPace is stimulating the brain to try to decrease the probability to have a seizure in drug-resistant patients. But this device is recording the number of interictal spikes per hour. So it detects them and it counts them. And it also counts the seizure so that the clinician then can download the information and get an idea of whether the treatment is efficient for the patient because they can adjust the frequency of the stimulation and so on. But at the same time, it produces a huge amount of human data. And this is the number of interictal spikes per hour. And this is the day, the night, the day, the night, the day, the night. And you can see there's a circadian regulation of the number of spikes. So the spikes occur in this patient mostly during the night. But what you see also on a longer time scale, that there's a number. So it starts, and the number of spikes increases, reaches a maximum, and decreases. Now, interestingly, seizures mostly occur nearly always during the rising phase of this rhythmic activity. Now, if I zoom out, so this was 2 weeks, and now we look at 2 years, and I get the envelope of this slow rhythm that you can see now here. So in gray are the interictal spikes per hour every day. And this is the envelope. You can clearly see the multidien rhythm over 2 years in this patient, which is ultra stable. And seizures tend to always cluster during the rising phase of this slow rhythm. So circadian rhythms are found in 95% of patients with epilepsy. It's not like a phenomenon that does not affect everybody. It affects everybody. These multidien rhythms in the patients equipped with a NeuroPace system, with a proviso that they have a system that stimulates the brain. It's also like 90% of the patients
they have this multidien rhythmicity. Each patient has its own rhythms. So most patients, it's around 7 days, and other patients, 2 weeks, 2 months, or a year.
Now, these multidien rhythms, there have been found, as I mentioned before, in 1938, they were already described. Now, if you take a diary, so patients have their own diary, and according to what the family is saying, they note when they have a seizure. They find also by just a human recording multidien rhythms. These are data obtained from the American neural-based system. This is the Australian system I talked about in the beginning, and they found the same results. You have dogs with epilepsy. This is occurring naturally in some dogs. They did EEG recordings in these dogs, and sure enough, you have multidien rhythmicity, and again, seizure clustering during the rising phase of the multidien rhythm. In rats, we have checked it, and we find the same thing. So across species with epilepsy, then there is a multidien rhythm. Now, what that is, where is it coming from? It's anybody's guess. So of course, it must involve species with enough lifetime to witness it. And we have started to look at control animals, because the question is this multidien rhythm a true biological phenomenon, or is it a consequence of epilepsy? So we used control rats, and I can give you just small indications. These multidien rhythms exist in control animals, and they are specific to each animal. So if you have 2 rats in a cage, they will have their multidien rhythm that is personal. So it's a true biological phenomenon. I have no idea where it's coming from. If you have some ideas, I would welcome them. But it's a new field. Obviously, it will be a new field of research in the future, just like the circadian rhythm. We will have to take into account the multidien rhythm. Now, that also may explain a few things. If there are multidien rhythms, if you use rodents, or if you use monkeys, for example, even humans, to assess cognitive function, for example. Even if you do it always at the same time during the night and day cycle, you always find a huge variability across individuals. And you have to have many individuals to get something statistically significant if you intervene in some way to do your cognitive testing, like a before and after a drug or whatever. But imagine that if we all have organisms, multidien rhythmicity, and we are at the peak of this multidien rhythm in terms of cognitive performance, so we will perform well. But the same, and another individual measured at the same time would be at the trough. So then you have a huge variability. So we tested that also in rats. And we tested every day with a very simple cognitive test, and the animal doesn't get bored doing the same test every day at the same time. And sure enough, the cognitive test shows that it's doing the multidien rhythm. So it's there, it has consequences, just like the circadian rhythm. You know that you learn better in the morning than in the afternoon. That's physiological. Right? You perform better doing sports in the afternoon than in the morning. This is regulated by the circadian clock, and in particular the suprachiasmatic nucleus. But the multidien rhythm has also similar effects. So following this slow cycle, you have periods during which, at least for rats, they perform better in cognitive testing, and other days they perform less. They're not stupid, they're just performing slightly better. So just to finish with the multidien cycle, it fits with the Moore hypothesis with this molecular architecture. Let's imagine that you have like a tide, okay, in the harbor, and the water rises at high tides, but it remains below the quay. Okay,
That's and the case the threshold to have a seizure. So every day it does this, and you get close to the threshold every day. But then regularly you have the equinox, it's called in English. Equinox? Equinox, yeah. So the equinox tide, where you get a massive surge, and then you go over the threshold. But it's every equinox tide, like it's interspaced in time. So that's the image that I would like to share. It's just hand waving, I have no idea, but that could be a possibility. So the final conclusion is that the circadian rhythmicity of seizures and interactor spikes is grounded in the basic properties of life on Earth, this circadian rhythmicity. And I don't think that it's limited to the brain. In fact, we have evidence now that all these oscillations also occur in the other organs. And since the other organs are coupled to the brain, that should be telling us also something about the system, but we don't know. These molecular oscillations, they give us cues to understand how to control now seizure risk or seizure probability, because we know when the seizure risk is high. So now that we know when it's high in patients, we could time our drug intervention to provide the drugs only when the seizure risk is high and not continuously, which is damaging to the patient as well. Of course, should we revise the way we study neurological disorders or any disorder in any organ by comparing health and disease? Is it valid? I don't know. Okay, now I'm going to shoot myself a bullet in the foot with the maybe the most important thing that exists for me in biology. The basic property for me of any biological organism is called degeneracy. I guess that you are familiar with that. Yeah. So degeneracy, it was first introduced for immunology, and then they derived it with the autonomy to the brain. And this is an example. So these are proteins. These are ion channels. And these are synaptic connections in the lobster. And these are the values of these ion channels. And these are the values of the conductance of the synapse, whatever. So they describe the internal state of a simple neuronal network in the lobster. This is the work of moderate complex. Okay. So when you have a network, a neural network made of these constituents, it produces an oscillatory pattern. Now, this thing here is totally different, right? So for example, even this channel is not expressed at all. But this channel in this network, it's highly expressed. But this network, which has a totally different constitution, internal constitution, produces exactly the same rhythm as this one. Okay? That's degeneracy. Degeneracy means multiple solutions to the same problem. Okay. What I told you is that when I look at the circadian map, it's changing from one moment to the next because of the circadian regulation of genes and proteins. So at one time point, if I want to generalize this, I have this picture. And later, during my life and day cycle, I have this picture because the genes have changed. The circuit looks totally different, but I have no idea. Maybe the circuit is producing the same output, but differently. So it's not really different. It's just the way it is made of, which is different. So it's using different solutions. Now, in epilepsy, what we always see that the system is totally different from the control. This is what I showed you. The circadian rhythmicity is totally different between the control and the epilepsy. But is it really meaningful? Because if epilepsy is like this and control like this, but degeneracy tells us it's the same output, maybe what I'm seeing here is irrelevant to my question. So it's very difficult when you do science to accept the idea of degeneracy, especially in the brain, because we want simple answers. But degeneracy is the basic property of any living organism of the planet. It explains evolution. It participates to evolution. It explains why several species, several members of a species can survive with a dramatic change in their environment because their internal composition allows them to do that. So instant evolution or instant survival, if you want. But degeneracy is not taken into account in neuroscience. I hope in basic biology it is, but that's the thing that complexifies the interpretation of our results. And just to finish with the last slide with the most complicated stuff. We study the brain because the brain is the most important organ in the body. That's what the brain says anyway. And neurological disorders, obviously, they somehow come from the brain. But in cell biology, if you're studying, I don't know, epigenetic mechanisms, okay, the writing of instructions on the histones, you're not going to say this is totally independent from metabolism. But we know it's highly linked. You cannot dissociate metabolism from metabolic activity from epigenetics in a single cell. That's stupid to make them two separate fields. It's part of the same thing. Well, the brain is part of a body. It's not alone. It's like a cell. The cell is made, you can distinguish several compartments and several things that are happening. But our body is made of many different organs, including the brain. And they all talk to each other. The vagus nerve carries axons, descending axons from the brain to the periphery, and there are axons coming up, picking up signals from the periphery to the brain. And in fact, 80% of the axons there go up. They go from the periphery to the brain. They tell the brain something. Until now, 99.99999% of the research is done on the brain, forgetting that there is something else outside the brain. And that's a big mistake. Just to give you an idea, if you want to hear fantastic talks, you should invite Catherine Talon-Baudry in Paris. She's a specialist in interoception, so how these peripheral signals are integrated in the brain. In humans, it's very easy. You put EEG electrodes on your belly, and you record the electric activity of the stomach. You record the electric activity that represents breathing, and you have the heartbeat. And you can extract breathing, electrocardiogram, the activity of stomach, and the heart. Okay, so you have everything. Now, what they have found, for example, is that in some brain regions, after each heartbeat, 200 milliseconds after, there's a field, change in the field. Now, they had a PNAS paper last year where they used patients implanted with electrodes and recording single neurons in the thalamus. And sure enough, you have neurons after each heartbeat, either they increase dramatically their firing frequency, and others, they decrease it. So after each heartbeat, you have a change in firing activity of neurons. Now, if you forget that there is something else than the brain, and you record these neurons, and you see, oh, every second is a burst of activity. So there is a pacemaker that produces this kind of rhythmic activity. Well, you're going to miss it completely. It's coming from the heart, not coming from a generator in the brain, or maybe it's in the brainstem, it goes back, I don't know. But forgetting about the other organs doesn't allow you to understand the brain. That's the point of view that I would like to make. So these are the regions which have been investigated so far in the human brain, which are sensitive to heart, to breathing, and to stomach activity. And you can see, like, most of them, because the others have not been investigated yet, the organs influence brain activity. What she showed also is that the alpha rhythm is based on the stomach rhythm, not the reverse. So a basic form of activity of the brain is faced by the stomach. Interesting. So just to tell you that we are far from understanding anything. These organs also show circadian and multidien rhythmicity. That's what we're going to show soon. But what I want to say here is that trying to understand epilepsy or any other neurological disorder, forgetting the rest, may be also a major mistake. These things, they generate a lot of electrical activity that we can measure, fortunately. But so far, we don't have at all a framework to try to conceptualize what is happening and how everything is integrated. But basically, the brain is always presented as the ultimate complex system where everything is coupled in a non-linear fashion and there are emergent properties. Well, now we don't want to forget that it's a complex system. The body is a complex system where everything is connected to everything in a non-linear fashion most of the time. And what we see is just a global emergent property from the body. And what we observe in the brain is an emergent property. That's my feeling of everything that is happening in the body, not only the brain. I want to leave you with this conclusion that is not optimistic. If optimistic, yes, because it means that we have now centuries of research in front of us. So we are far from understanding anything. But there's hope for the new generations because these are very beautiful topics that remain to be investigated. Thank you.
RNA data that you showed where you have the downregulation and the upregulation of the control versus the epileptic. It'd be interesting to see if you took somebody and you gave them severe jet lag, that you moved them to a completely different side of the planet and see what happens. Do you get a recovery of what those normal kind of up and downs were for those things? And could the control maybe recover faster than the epileptic brain? Are there ways that an epileptic brain maybe would not be able to adjust to changes in circadian rhythms as quickly? Would that maybe mean that there is maybe lags in the circuits between those different protein pathways or molecular pathways that cause them to have kind of a different dynamic during different time periods? But you have to compare that, to itself, right? So what you had before the jet lag versus afterwards.
So this has been done in animal models, cannot do it in humans. But in animal models, if you switch, if you introduce a jet lag, there's a phase reset that takes, depending on the duration of the lag, 2 to 3 days, and it goes back to the new second pattern.
And there's no difference between how a control animal adjusts and recovers versus an
This has not been compared. Now it's more complicated than what I presented. These two science papers, they did a fantastic job looking at what is important. The circadian clock controls the transcriptome, but the sleep-wake cycle controls the proteome. So the circadian is like sex, okay? And that's the transcriptome. Now the proteome is controlled by the sleep-wake cycle, which is that if you perturb sleep, you won't have an effect on the transcriptome, but you will have an effect on the proteome. Just to make things more complex.
So if you wanted to twist some knobs, let's say that an animal is going to have a propensity for a seizure at a certain time. Maybe by adjusting the sleep-wake cycles of that animal, which may possibly change whether or not that epileptic seizure occurs, or even maybe light exposure. If you could extend the light exposure and cut it off at different parts of the day, kind of create an artificial jet lag for the animal.
So that's a very good question. This has been done. This is not in humans. If you increase the quality of sleep you will decrease seizure risk. If you have a bad sleep, it increases seizure risk. So there are some kind of intervention they can bring, including using drugs that normalize in a way sleep, and that improves epilepsy. That's clinical data. There's not much in terms of animal research on that. Now, whether if you jet lag them in an appropriate manner, you would have to jet lag them all the time, so that you shift them back to the low seizure risk period. That's a very interesting idea, but my guess that you're going to perturb sleep so much that it will have the detrimental effect, because that's sure. Even in animal research, it perturbs sleep in an epileptic animal, they will seize more. Well, that's a good thing. Now,
Is circadian rhythm just light duration, or as far as like light duration on, off,
now
the gradient, or is it intensity? Because if you could change it with like an intense light therapy as well, right? So you get like 10 minutes of intense light therapy.
so in humans, they tried the 40 hertz stimulation, because that's the gamma rhythm, but there's no very good outcome. Doesn't seem to have. The trials that have been made are inconclusive. They tried it in Alzheimer's patients, but so far we don't see something dramatic coming out of that. But now the cool thing that we have the Multidien rhythm is that we can predict it in animals. So we can now time, when I told you at the very beginning that it's impossible to know when the seizure is occurring, and we would like to see what the circuit looks like just before seizure happens. Okay, that could give us some information. But now we can do that. Not when before the seizure is happening, but when the seizure is super high, that the risk is super high. It's in the Multidien curve in the rising phase. So we know when to kill the animal and do transcriptomics, for example, and compare it to another time where it's very low risk. And that way, epilepsy becomes its own control. You don't need to compare it to a control animal. Epilepsy is its own control. It's an internal control. Now, in science, when the system that you're using is its own control, that provides you with the most powerful tool to investigate mechanisms. But in epilepsy, we can do that. Not in the same animal, because we can cannot kill the animal twice. But maybe if there are fundamental rules behind these Multidien rhythms, whichever their rhythmicity, then maybe we can try this. That's what we are doing now. We've got an NIH1 to just investigate that.
Do twins have the same biorhythms?
Multidien, we don't know, because that's never been studied before. I don't know. So just to go back to the one question I didn't address, the brain has an autonomous clock. So the suprachiasmatic clock has an autonomous clock. It's autonomous. So if you people, the experiments have been done to put people in the cave in the dark during weeks. And the experiments have been done also in the South Pole, where they were staying like in constant daylight. And you don't have the everyday synchronization when the light is on. And our clocks are not 24 hours, they are 23.5 or 24.5. And each individual is going to switch. Going to switch to light or to drift. Okay. But every day in the morning, when that light is on, that resets our clock, or when we get food, it's a slight yellow. It resynchronizes the clock. But otherwise, the brain has its own autonomous clock. The stomach has its autonomous clock. The gut has its own clock. So if you put them into a dish, they will continue to oscillate with their own period. Now, each individual has its own period. It's called free running period. So when you put them into the dark,
people will carry on drifting with each other in terms of sleep wake cycle. So,
Do you know if patients who have had many seizures, can they like feel when they're going to, when they're at risk of them?
yes, some patients can tell the clinician, and the clinicians will not believe in that. I'm going to have a seizure in 3 days. And it happened to me already 4 days ago, and it's every 7 days. And the clinicians don't believe patients. But it is like magic. Like, oh, you can predict the seizure, but nobody, it's been searched for by brilliant minds during decades, and nobody could do it. And you, a patient, you can predict your seizure? Come on. So they were not listening.
It's a very long observation on patients.
Well, like,
It's
dogs,
not-
yeah,
but
dog-
instead-
dogs can smell it. things.
Exactly, but is that based on the empirical observation that it comes every week, or do they actually sense something?
So, some patients sense it. But it's usually the next day. They say, I'm going to have a seizure.
I was also wondering if you're agnostic of the time period, do all epileptic patients show some sort of rhythmicity in their events?
95%.
95.
95% of drug-resistant patients, because the others who are controlled, you don't know. Because they don't have seizures.
But that might give you an indication of what systems are actually driving. Are causal. If you know the time period, you can look for systems that actually act in that same interval,
Yeah.
which might give you some indication.
Well, there are zillions of parameters. So which one are you?
Yeah, no, for sure. But I mean, in the end, I guess that's the direction you should be looking at.
Yeah, that's what we are trying to do.
And what
But
have you considered moon cycles?
Some patients are regulated by the moon.
Yeah.
They are called werewolves. But like, it's like some, not all clinicians practice interrogation, clinicians listen to interrogation, but when they say something like this, they are very doubtful because it's non-scientific. They think that it's a secret, but magic. It's a very small number of patients who are like this, but they are not. Clinicians know that. But it's like a magic idea or a dogma. Now, there was this idea that women were synchronizing their cycle by the moon, by the full moon. That's stupid. It's not science. Guess what? It's true. It's true, but for women living outside civilization with no strong heights during the night and living on with the sun, and they are not synchronized for every full moon, only a specific full moon when the moon is closest to the earth, because the trajectory of the moon is elliptic, and at some time you have the big moon, and that's when they get synchronized. And that's a beautiful E-like paper. What they showed us, they get synchronized by this type of moon when the moon is closest to the earth. So there are things that we still don't understand. There's some knowledge that is based on observations that may be millennia old that we don't consider because they look like non-scientific, but these are observations from people. The Babylonian tablets 4,000 years ago already described circadian patterns in patients with epilepsy. It's on the Babylonian tablets. It's there. It's been described long time ago. So common knowledge may be a source of intuition for science. But we don't know. Yes.
I was wondering, we showed those brain activity patterns and how they seem to the high sensitivity for the epilepsy premises. is the max amplitude modulated or treated by the frequency of the sound. So as you mentioned, some patients have 2 days, 5 days, and other ones 30 days. But for those two patients, is the max amplitude the same? The number of seizures and what's high rising? It's the same.
No, it's just the rising phase that correlates with the seizure probability. And in fact, it's a double curve. So you have the multidien plus the circadian. And when you cross the two, you get maximum probability, maximum risk.
So that high rising period is not higher or lower in patients with not much.
Do you know, like having intent as to what point multidien as to be
That's a multi-billion-dollar question. So I asked specialists I know of hormones. It's okay, maybe it's hormonal. The cycle that we know best is the woman's cycle. So they told me, well, the hormone field is still to be discovered. We knew a few hormones, but the others, we have no idea what they do. So many have been identified, but research is still pending. So I'm thinking that maybe there's a hormone that is released that produces this rhythmicity. But then I talked to the last student of Degeneracy, the Nobel Prize physics, and he's working on systems at the limit of thermodynamic equilibrium, which is living organisms. And he looked at the interaction between the cell cycle, division cycle, and the metabolic cycle. And he made a big model that explains the relationship between the two. And just these two oscillatory things, when you couple them, you have a very complicated system. And so that's why I contacted him and said, well, there's a very simple way to explain your phenomenon. And I didn't think about it. It's obvious. There are two oscillators, and one is slightly shifted in time compared to the other, phase shifted, and that produces a slower oscillation. Now you have oscillations everywhere. You have oscillation in the stomach, in the heart, in the respiratory system, in the gut. They all have this quasi-circadian rhythmicity. Now, if you put them all together, maybe you have like multidien rhythm that is generated because you have less coupled oscillators. That could be as simple as that. No molecular events, no biophysical mechanisms, just a coupling of oscillators. It could be as simple as that, but I don't know how to study that. Right. But it's a very nice hypothesis.
One question based on the heartbeat growth responses. How do those look like in people with a heart rhythm? So they are 3 normal heartbeats, or just every 2 or 3 heartbeats as an extra one? Wouldn't that like cause like havoc in the brains?
So
they seem pretty asymptomatic.
So as you understand, doing single neuron recordings in patients is extremely rare. And you can do it only in patients with epilepsy because you put electrodes in their brains to diagnose their epilepsy. Now these bits, they have small wires that you can push from the skull, and they will record something else. So you cannot put your electrodes anywhere you want to record. It's initially puts the electrode there, and then you happen to have some neurons recorded at the tip. So there's a very limited number of recordings, and I don't think that they have any single case for that. So they use what they have. It's already extremely rare, and also very interesting to have these recordings when you can get them back. But it could be done in line. In fact, I have a big project now recording all these organs and the neuropixel probes to see what is happening in the rodent brain, and playing with also with pharmacology to alter heart function or
the stomach and see how it modulates brain activity. Just right now, observation, no theory, no hypothesis, just let's see what happens.
You said that reset can be done, yeah, so, and mostly it's vagus regulation, some drugs, yeah, are epileptic drugs, also targeted vagus, so it's kind of reset this vagus. Yeah.
It could be, and in fact, one clinical device is vagus nerve stimulation, and that was maybe the first one introduced as a complement to neurosurgery, because some people cannot go through neurosurgery because you remove eloquent cortex, for example. And so they had this device where they have a clamp on a vagus nerve and they stimulate constantly the vagus nerve. It helps some patients and some patients doesn't do anything, but it clearly shows that by acting on the ascending or descending pathways, we don't know which ones are activated, you can change also seizure probability.
So basically this resets through the organs like lungs, stomach, that can be also through, can be through vagus, yes.
Thank you so much.
Try to keep it in euro.
Yeah,
So that-
no, perfect. So can we talk for a little bit?
Yeah,
Sure, yeah.
yes, I would love to, because I need to